is harmful. This can be infections, allergies, and also the autoimmune disorders when things go a little bit out of control. The symptoms of inflammation really vary with the severity and the trigger, but can include chronic pain, redness, inflammation, swelling, stiffness, for instance, of the joints. And really, when things go a little bit out of control, you can have damage to the tissues. If you look into inflammation, you can basically classify it in type 1 and type 2, and that kind of relates with the type of T cell population that is involved. So if you have a response that is triggered by a T helper cell 1, then you have a type 1. As most things in life, I think, you need to have balance. If you have too little inflammation in response to a trigger, you can clearly lead to tissue destruction. Let's say if your body is not able to fight, for instance, an infection of the brain, you may end up with a damage of brain tissue. On the other hand, if you have too much of it, then you can also lead to tissue destruction. And one example is osteoarthritis. So the body has developed very tight mechanisms to keep things in balance. If we talk about inflammation, this is a very broad term, and there's a number of diseases which are debilitating and damaging to the individual that link to this inflammation process, and they affect a number of tissues in your body. So if you think, for instance, of the head and neck, you can develop encephalitis, conjunctivitis, sinusitis, meningitis. Then you have infections and inflammation of the skin, dermatitis, lupus. Although lupus tends to be really linked to the symptoms shown on the skin, in truth, it is a disease that affects many organs. You can then have affection of the lungs and respiratory system. You can develop emphysema, bronchitis, asthma, digestive system. You can have colitis, pancreatitis, IBD, Crohn's disease, celiac disorders. You can have muscle and joint disorders such as RA, osteoarthritis, and also in very damaging and lethal, you can have problems of the heart and kidneys, such as pericarditis and myocarditis. So as you can see, under the umbrella of inflammation, there's a very large number of disorders and diseases. And so this is really a field that has been highly investigated, and it's highly relevant. Just an example of a disease, just to give an idea of how frequent and how severe inflammation can be. One of the diseases under this umbrella is COPD, which basically means chronic obstructive pulmonary disease. And this is a disorder that affects about 5% of all the adults. You may not think about it, but it's the third leading cause of death. The causes can be smoking, secondhand smoke, lung irritants, genetics, air pollution, and alpha-1 antitrypsin deficiency. And the cost, this is an estimate from 2010, so obviously this has risen, but in 2010, the cost of COPD treatment was $32 billion. So this gives you really an idea of how big the field is and how important it is to continue investigating and investing in this field. I won't go into all the details of this figure. I just wanted to give an idea of how complex inflammatory responses can be. So for instance, if you have the epithelial of the lung, and this is hit by allergens or virus or bacteria or cigarette smoke or pollutants, there is a trigger. The cells are going to secrete molecules, one of them being TSLP, which then will affect other cells, which will then release cytokines and in turn affect other cells, and so on and so forth. So this is really a cascade of signaling pathways that are activated, and it's highly complex. So the more we understand these pathways, the easier it's going to be for us to develop strategies to fight them. And I want to highlight in the scheme that you see a lot of the TSLP. So that's really a molecule that is being under investigation and is revealed to be a crucial factor in this response. Now I just wanted to introduce to you what we believe and how, I wanted to introduce to you how at BPS we think about the discovery workflow and how we develop our products, and then link this to the presentation. So we think of research in stages. You start with a stage where you're thinking about your target protein. And for that, you're going to develop and need some reagents, such as recombinant proteins and antibodies. You then move to the next stage in your research, which can be, for instance, compound screening and lead molecule identification. And at that point, you may use biochemical assays. You may need inhibitors and activators. Then you're going to identify your lead molecules. And obviously, before you go into animal studies or any clinical trial, that research has to be validated in a cell-based model. It's a more complex model, is one that mimics more the in vivo situation. So for that, you're probably going to need cell-based assay kits, cell model, cell media. Finally, before you embark into a clinical trial, you want to do a verification, if possible, and available in an animal model. For that, we can support, and you're probably going to need a cell line, a reporter cell, maybe a viral-based tool. And this is how we developed our tools at BPS to support every stage of the research. So I'm going to introduce to you during the presentation tools that fit into every one of the stages of discovery. I want to spend just 1 minute or two in terms of the cell culture workflow, because there's also links to how at BPS we develop products. Basically, when you purchase a cell line or you have the cell line in your lab, you have them probably as a frozen bank. You're then going to thaw it. You expect and hope your cells recover well. You have cell viability at a good number, and your cells are in good shape to expand. You're then going to have to expand them to a certain number. Either you need very little amount of cells, or you're going to have to scale them up into really large numbers. And at that point, typically you're going to create a cell bank for future use, and you're going to perform your assays, either drug development or cell-based assays, immunofluorescent, flow cytometry, whatever the assay may be. At BPS, we try to develop media that fits into every stage of that workflow. So we have media that is the thaw media. This is a media that has been optimized for the thawing of the cell line that it relates to. As I mentioned, then you're going to grow and expand your cells. So we have developed a growth media. And then if you want to perform assays, we have an assay media, and to freeze your cell bank, a freezing media. And you may think, why do I need to really invest in purchasing the media from BPS? Why does every data sheet of a cell line come with specific media formulations? Well, the reason is that each media that we've developed has been optimized, characterized, and has been validated to work as an optimal media with those cells. So we do recommend customers to purchase them. We do have the questions often, why do I need to buy your RPMI with FBS? Well, because we have validated the specific source of RPMI, and we have validated the specific FBS to be used. And if you have done cell culture for quite a while, you are very familiar with this triangle interfacing each other, where for you to have a desired cell properties, you do need to play with the media you're putting in and the growth conditions. So we do recommend people to purchase our media and follow the directions that detailed in the data sheets. And we do put a lot of effort into those to provide you with the best reagents and the best protocols. And in turn, we hope the cell lines perform as they should for you. Going a little bit into the TSLP and TSLPR. So as I mentioned, when you have, for instance, the epithelial in your lungs, the cells are then almost on a daily basis bombarded with allergens, chemicals, microbes, and all the other insults the cells suffer. And in response to that, they produce molecules. And one of these molecules is TSLP. This is produced by epithelial cells in the lungs, but also in the gut and dendritic cells. Once that molecule is secreted, it's then going to act on, for instance, immature dendritic cells, but also CD4 positive cells. This then will release cytokines and chemokines that will trigger cascades in other immune cells, and then you end up with a response that is inflammation. So, for instance, this can lead to mucus production and tissue rearrangement, which closely links with COPD. If you remembered, COPD can be characterized by emphysema, but can also be characterized by remodeling and mucus secretion. TSLP has multiple functions, so it can function as a cytokine, it can function as alarmin, and as a growth factor. I find the name alarmin quite interesting and fun, and I think it really links a lot with TSLP because basically it's working as an alarm system for your body. Once there is an insult, the cells release this alarm molecule, which then is going to trigger other reactions. Diseases that relate to TSLP include COPD, allergic reactions, any autoimmune disorders such as psoriasis, RA, and cancer too. The signaling pathway really starts with TSLP binding to TSLPR. TSLPR is one of the subunits of the receptor. The other subunit necessary for intracellular signaling is the subunit that is shared with IL-7, and it's the IL-7 receptor alpha. Once this subunit is recruited, you now have the complete receptor, and you can then trigger the intracellular pathway. Basically, you have activation of the JAK and STAT signaling, which then will trigger transcription of cytokines involved in inflammatory responses. So internally, there is a lot of signaling happening that is not represented in this figure, but I would ask you to keep in mind this generic figure for a product I will introduce to you later. This protein is also linked to cancer. Its role in cancer seems to be a little bit under debate. Some studies describe it as an oncogene, some others seem to be describing it as a tumor suppressor. It seems and it's highly possible that this relates to the tumor cancer type specific, but it is known that in some solid tumors, this molecule can affect tumor microenvironment and lead to cell proliferation and metastasis. So really a lot of roles which can be highly detrimental if not kept under control. As I mentioned, this is a very interesting field, and due to the umbrella of diseases that is under inflammation and also the number of individuals affected, there's been a lot of research going on in the last couple of years. And so there's a lot of targeting strategies that have been developed more or less recently. And those include neutralizing antibodies. So basically, you develop an antibody that is going to bind to TSLP, and then this does not allow the TSLP to bind to its natural receptor anymore. You have what is called cytokine traps. So basically, you're trapping the TSLP. Now it's in a conformation and in a molecule that does not allow the penetration into the receptor. You can mimic the receptor in a soluble form. So if you think about it, it's now soluble. It will not be able to trigger the intracellular pathways. So that's a very clever way to stop the signaling from happening. You can also use mutated cytokines. So basically those will mop the receptor for you and will not trigger the right signaling pathways. And then you can have small molecules or peptide antagonists. And the result of all this really is that you want to stop the intracellular signaling from happening and the disease from developing. As of 2020, so this is a little bit outdated, but as of 2020, there was a number of clinical trials ongoing. I believe I consider them quite successful. For instance, the use of this specific antibody, which is a human IgG2 monoclonal antibody, tissupilumab. From what I read, subcutaneous application of this antibody for 52 weeks did lead to a reduction in 62% of episodes of asthma that led the patients to the hospital. So I think this is really life-changing for these patients, having to deal with the asthma episode, having to go to the hospital, being under all that stress is really detrimental to life quality. So I want to point out from this table too that the preclinical studies for this specific antibody were done in this cell line called BAF3. And so this is a mouse cell line that was used for the preclinical studies. And I'll bring this later on to you. But as you can see, there's a number of molecules being studied. Like I mentioned, cytokine traps, small molecules, monoclonal antibodies, and so on. Bringing you now to some of the reagents we can help you with and support your research with. We offer a TSLP responsive luciferase reporter BAF3 cell line. So a bit of a full mouth of a name, but it is a mouse cell line. As you can see, is a BAF3. This is why I wanted you to keep in mind that the cell line was used for the preclinical studies of the antibody. Cell line has been validated, for instance, with anti-TSLP neutralizing antibody. So it does contain a luciferase reporter. So when TSLP binds to the receptor, it then leads to the activation and expression of luciferase. When you incubate it with a TSLP neutralizing antibody, of course, you're going to see a response and a decrease in luciferase activity. As I mentioned, we do provide the media. So this product is supported by a specific thaw media, a growth media, an assay media. And in addition, we can help you by providing you with a mouse interleukin-3, which you will need since the BAF3 cell line is dependent on this interleukin. And we can provide you also with a neutralizing antibody, which you can use as control for the cell line performance or for the development of new tools. And our very successful product, the one-step luciferase assay system that we recommend using for luciferase activity. We also offer recombinant proteins. So if you remember from the workflow, you start by an interest in a specific molecule. So at that point, you probably haven't developed all your assays and tools. You start with the need for recombinant proteins. So we wanted to support you in that. So we offer TSLP, also TSLPR, with different tags. Our proteins are all high purity proteins and are validated by SDS page, and when possible by biological assays or biochemical assays. Proteins are biotinylated, such as our 102, 169 are also validated by evident pull downs. We offer multiple sizes for your convenience. And also, as you know, when you increase sizes, you tend to have a better bit of a pricing. So also for your savings. To help you develop your tools, we do offer you anti-TSLP neutralizing antibody. This is a recombinant antibody, so it's not a polyclonal derived from rabbit or derived from hybridoma. It's a recombinant monoclonal antibody. This provides you with the advantages of having minimized lot-to-lot variability and also a higher reproducibility of results. This antibody has been validated in a cellular-based assay in addition to STS-PAGE, so you can see that this antibody is really able to neutralize and inhibit the responses. The next stage in your research, when you probably have or decided which molecules you would like to test, maybe the need for an assay kit. So we have developed a very simple to use assay kit that allows you to measure the binding between TSLP and the subunit TSLPR of its receptor. So it's a inhibitor screening chemiluminescent assay kit. So really very easy to use. It's basically an ELISA type of assay. You start by coating your plate with TSLPR. You're going to block it. You then going to add your molecules of interest, either be a small molecule or an antibody. And I want to mention that these assays have been validated with both types of molecules. You're then going to add your TSLP biotin, streptavidin, HRP, finally ECL substrate, and then you're going to be able to measure the chemiluminescence. So obviously, if you block this interaction, you're going to have a lower amount of chemiluminescence. So very easy to use. I think every researcher is basically familiar with ELISA type of assays. So we wanted to make it very convenient, very easy, very straightforward. This allows for the assessment of the inhibition of blocking of TSLP binding to this subunit of the receptor, either by antibodies or small molecules. It has been validated with anti-TSLP neutralizing antibody. Again, it's a very straightforward assay, very convenient. The readouts are quite sensitive, and it allows you to compare molecules, potency of different types of molecules if you're interested in developing new ones. And we do recommend that if you use our assay, you do use the anti-TSLP neutralizing antibody, since this is the molecule used as development molecule. Talking a little bit more about the cell line that I mentioned in the beginning that I wanted to highlight to you, but just to reinforce, this BAF3 is a mouse B cell line, is IL-3 dependent. This will be the product basically that you're going to need when you want to validate your molecules or drugs in a cell-based assay. The way this was engineered, we basically had to introduce both TSLPR, but also the IL-7 receptor alpha subunit. If you remember, for you to have intracellular signaling, you're going to need both subunits. Although the cell line is a mouse cell line, the proteins introduced are human, and we have validated that the signaling pathway is active. Finally, the cell line uses a SET5 luciferase reporter. Once this is activated, it's then going to lead to the production of luciferase, which you can easily measure with chemiluminescence. The cell line has been also validated by flow cytometry, so we have verified the expression of both TSLPR and IL-7 receptor alpha, as you can see in the flow cytometry graphs, both in green and red. And in addition to the anti-TSLP neutralizing antibody, obviously the cell line has to be responsive to TSLP. So we have verified that. If you include TSLP in your media, you do see a dose response curve. Very nice. It's very sensitive, so you can have a full induction of response of 0 to 100. So this really allows you to measure small differences in potency of molecules. Again, we do offer the tools you're going to need to work with the cell line. They've been optimized. They are available for you. And I just want to wrap up this portion of the presentation by saying that we hope to provide you soon with a human cell line expressing TSLP, TSLPR. Sorry, I meant TSLPR. Although that is a step forward versus the use of a mouse model. As you've seen in this presentation, the use of the BAF-F3 has been used in preclinical studies, so it is a valuable tool. We just wanted to provide you with even a better model, which would be a human model. Changing a little bit gears and moving to a different molecule also involved in inflammation. So I think you're probably all familiar with TL1A, DR3, and DCR3. And this is also a very interesting type of molecule. And in fact, all the molecules involved in inflammation all have their little interesting mechanisms of actions and their little niche actions. So TL1A belongs to the T cell co-stimulatory TNF-RSF family of proteins. So again, a very large name. It's produced by a number of cell types. So for instance, it can be produced by dendritic cells, macrophages, but also cells that are not really immune cells, such as the synovial fibroblasts and endothelial cells. Once this molecule is released, it's then going to act on other cells, such as naive T cells and NK cells, which respond then by producing chemokines and inflammatory cytokines. And again, is linked to a number of inflammatory disorders, such as RA, IBD, psoriasis, lupus, and other types of disorders, all of them highly debilitating and that have an impact on the quality of life. The signaling pathway of this molecule is slightly different from the previous one. As I mentioned, each of them has their own little niche in specific signaling tricks. So basically, most of the TL1A is found as soluble. This protein is produced and then is cleaved by this molecule called TAS, and it then becomes a soluble molecule. The receptor present on the cells is called DR3 or death receptor 3. So once the TL1A is going to bind to the DR3, this is going to activate signaling pathways. And depending on the signaling pathway that is activated, you end up with a different outcome. For instance, if the activation leads to recruitment of TRAF2, this end up with the outcome being inflammation, differentiation, proliferation. However, if then TRAD is going to link to a different type of molecule named FAD, this can lead to apoptosis and in extreme cases to necroptosis, which is really a high stage of inflammation. So as you can see, depending on the pathway activated, the outcomes can be slightly different. And the interesting thing is with TL1A is that the body has developed mechanisms to keep this protein under control. And that was done in a very interesting way. So basically, we have in our body something called DCR3, which is decoy receptor 3. And this is a soluble receptor for TL1A. So if there is excess of TL1A and you don't really want to lead to extreme inflammation, you have this molecule that is going to capture it, and because it's soluble, is really not going to allow the intracellular signaling. So interesting, you can have a molecule that then is going to act as an immunomodulator, and by using DCR3, you're going to inhibit apoptosis and reduce inflammation. So this is our body trying to find mechanisms to keep inflammation under control. And at BPS, again, we have developed recombinant proteins with multiple tags. They have been validated either by STS page. We also perform SEC, and when possible, biological and biochemical assays, such as in the case of the TL1A is tag-avitag recombinant. You can see here a nice cell-based assay where you can see a very nice dose response to exposure to TL1A. This is high purity proteins, multiple sizes available. we are very open to work with you to provide different tags, different purity levels, different sizes. So please just reach out if you have any question. We have developed a very similar type of biochemical assay that measures the binding between the intracellular receptor, the DR3, and the TL1A inhibitor screening assay kit. So again, it's an ELISA type of assay, very straightforward and easy to use. The applications are the assessment of the inhibition or blocking of the binding of these two proteins. It has been validated for the use of small molecules, but also antibodies. So we have put the effort into developing specific protocols for each of this type of molecules, and it's been validated with the anti-TL1A neutralizing antibody. I won't go over the scheme because you're all very familiar with the ELISA type of assays. In addition, because of the high interest that there is in the capture of TL1A with DCR3, we also developed an assay kit that measures specifically the binding of these two proteins. It allows you to assess the inhibition of blocking of this binding. So in the case you do want to activate your pathways and block the binding of these two molecules, you can use this kit for your testing. You can see here that this was validated again with our anti-TL1A neutralizing antibody, and you can clearly see when you introduce this antibody, you do see a decrease in the response, meaning that the DCR3 is not able to bind the TL1A anymore. This is our antibody. We believe it's a very good product that can be used in multiple applications. You can either use it for studies where you're interested in studying the binding of TL1A to DR3, but again, you can also use it for the DCR3 TL1A, both as a control or a tool for manipulation of the pathways. Finally, in your last stage of development, you're going to need a cell model. So we have developed a TL1A responsive luciferase reporter Jurkat cell line. So in this case, we do have a human T cell line, the Jurkat cell line. This is a very common model in immunotherapy studies. What we've done was we introduced the DR3 into the cell lines. Then there is a activation of the signaling pathway. And finally, you have activation of NF-kappa B and luciferase expression. So again, a very simple model where you can have a nice outcome and a very easy readout of your studies. Once TL1A binds to the receptor, it then triggers the signaling pathways that lead to the activation of luciferase expression. In addition, the cell line, although you are seeing the DR3 in the scheme, it can also obviously be used for studies on DCR3. Basically, you have to introduce DCR3 into your cell media, and then you can check if TL1A is being captured or not by measuring the luciferase. The cell line has been validated in multiple ways. So we do our best at BPS to provide tools that have been extensively validated, obviously within our limitations, as we don't have access to all the drugs out there. Many of them are patented, but within possible, we do our best and we try to validate our tools as much as possible. And if you have any request or you have any question about a specific type of validation, this is something we can possibly work with you. So feel free to reach out. But this cell line has been validated in a couple of different ways. One of them has been the incubation with TL1A. Obviously, for the cell model to be of any use, it has to be responsive to the TL1A. So if you compare the response of a cell line, which is our NF-kappa beta luciferase Jurkat cell line that does not express the DR3 versus the cell model, you can clearly see a dose response only in the presence of the DR3. And we do recommend that a control is used. It's good experimental practice to use as many controls as possible. So a cell line that does not express DR3 but does have the remaining of the machinery is a very useful control. We also wanted to test the specificity of the cell line in response to other molecules. So we have chosen TNF alpha. Again, you can see that the response of the cell line is very specific to the TL1A. And we have validated it with our anti-TL1A antibody, and you can see a very dramatic response once you incubate the cells with this antibody, and luciferase percentage of luminescence really goes down to almost zero. So it's a very good and sensitive module. We also validated again, as I mentioned in the beginning of the cell line, this cell line is also useful for studies that involve the DCR3. So basically, if you incubate the cell lines with DCR3 and you want to study, let's say, a TL1A molecule or an antibody, you can have an indirect response. So in this case, what we've done is we incubated the cell line with TL1A, and you have to have the stimulus present to see a decrease in response. We've then added DCR3, and you can see very clearly a very impactful response by losing almost all the percentage of luminescence. We have developed specific media, growth media, assay media. We do provide a number of proteins you can use in your studies. So you're going to need TL1A, obviously, to stimulate your cells. So we can provide you with that. If you wish to have the control, we have the anti-TL1A antibody. In addition, if you need negative controls, we will recommend to use TNF alpha recombinant protein, so as a negative control for specificity, and also our NF-kappa beta luciferase reported JERCAT cell line, as you really want to make sure that the responses you're seeing are coming from the TL1A. Coming soon, we want to offer you with growth-arrested TL1A responsive luciferase reported JERCAT cells. So really the difference between what we're offering now and the new product is the cells are going to be growth-arrested, meaning they cannot divide. This is, we believe, a tool for customers that are interested in doing quick assays. You know, they're not interested in expanding cells or spending their time growing them. They just need cells to run an assay, and it will come with the advantage that it will not require a cell line license. So I do have to mention that our cell lines are linked to a cell license agreement, which we understand may not be ideal for some of our customers. So we do want to provide with alternative options, such as the growth arrested option. Finally, going to the OX40, another interesting molecule. And although in this presentation, they seem to be disconnected from each other, if you remember the first scheme, really the response to inflammation is a multi-process, multi-molecule type of response. So they really all interlinked. Is where you want to act that really makes the difference. So OX40 belongs to the T-cell co-stimulatory, again, the same family of proteins as TL1A, the TNF RSF family. This one is an immune checkpoint, so it's slightly different. And it has a ligand, which is called OX40L. It's the ligand, and can be found in multiple immune cells. So if you look at the table, you can find them in T cells, Tregs, dendritic cells, B cells, and they express either the OX40 or they express their ligand. In response to that, when the ligand binds to the OX40, you can have inhibition of T cell apoptosis, you can have promotion of T cell activation and proliferation, inhibition of Treg, so on and so forth. So again, the same similar type of mechanism, once the molecule binds to the receptor, is going to activate the signaling pathways in immune cells that can lead to cytokine secretion, and the cytokines will then act on other immune cells. Unfortunately, this is a molecule that has been linked to the cytokine storm syndrome, which, as you're probably familiar, is one of the potential side effects of CAR T. So really, it's a very important molecule to keep in mind and control when we're thinking about this type of immunotherapy. The signaling pathway, again, is slightly unique. You have OX40 is going to bind to OX40 ligand, which is present in antigen-presenting cells. And this in turn is going to trigger an internal signaling cascade. The signaling cascade is dependent on the recruitment of a protein called TRAF2. This forms a complex. This complex then is going to work and lead to the final action, which is the activation of transcription of cytokines involved in proliferation, survival, or inflammation. This molecule actually has two types of mechanisms. It can respond to TCR signals, the T cell receptor signaling, but it can also be independent of the T cell signaling. So basically a multitude of actions that result really in the end, which is the outcome being production of cytokines and chemokines that will trigger other signaling pathways. It's involved in a number of diseases, lupus, RA, multiple sclerosis, and a disease that in my mind for a very long time I've never really linked to inflammation, but is type 1 diabetes is actually an inflammatory type of disease. And then you have myasthenia gravis and some more rare disorders such as Graves' disease. In terms of therapeutic modalities, because this molecule is also involved in cancer, there's actually been interested in developing therapeutic modalities that both cover cancer and autoimmunity. And it's very interesting because if you're thinking about cancer, you really want to activate this molecule and you want to activate the signaling pathways involved and are triggered by this molecule. So if you think about that way, the type of drugs you want to develop and study are molecules such as agonist antibodies. You want to introduce soluble ligand into the cells. You may want to do bispecifics that are agonist antibodies with blocking of other immune checkpoints. You may want to do combinational therapy, but really is all about activating and pushing the pathways of OX40. But when you're thinking about autoimmunity, you actually have to think the other way around, right? You want to stop the cells from being so active. So in that case, you want to go investigate blocking antibodies. You may want to think about having soluble OX40 and that type of tool that inhibits the signaling. So really very interesting how you can use the same molecule in totally different ways. And I just mentioned this, sorry, this is, I wanted to show here the type of tools that have been used. So you can see that you have humanized IgG1 agonists, but you also have bispecifics, lipid nanoparticles, you have Fc fusion proteins, and so on. So really different types of molecules and different types of approaches that have been used. There is a large number of clinical trials ongoing around OX40. That doesn't mean that there's not more to study and more to develop. So far, it seems really promising that the use of combinatory therapy. So you want to probably not just use a target against OX40. So for instance, it has been shown that the use of OX40 monoclonal antibody combined with a different type of strategy, such as adoptive cell therapy, led to a regression of tumor sarcomas from 9 to 70%. So this is really striking. Although this was a mouse model, it's still a very striking result. So the pathway forward may be really the combination of multiple types of therapies. And at BPS, we provide you not only with the tools for OX40, but you can also look into our portfolio and you're going to find tools for PD1, PDL1, and other types of molecules that can be used in combinational therapy. We do provide recombinant proteins. In this case, we do have an Fc fusion, an AVITAG, ISTAG. In addition to human proteins, we also offer you mouse proteins. So you can see they're high purity proteins that are readily available for use. We have two types of antibodies available for you. Both of them are recombinant monoclonal antibodies, so minimal issues with lot-to-lot variability, high production yields, and so very cost-effective for you. We have a competitive antibody and an antagonist antibody that you can use. And we also offer an anti-OX40 antibody that is PE labeled, so you can usually very quickly to determine the expression of this molecule in your cells by either flow cytometry or immunocytochemistry. We offer an assay kit very similar in design to the previous kit. So again, very easy to use that allow you to determine and assess if you can inhibit or block the binding of OX40 to OX40 ligand by either molecules such as antibodies or small molecules. And it's been validated with our OX40 neutralizing antibody, and you can see a very nice dose response. So if you're interested in developing strategies that inhibit or boost the binding of these two molecules together for either application in cancer or autoimmune disorders, this is a kit that can allow you to do high throughput screening in a very quick and sensitive way. Moving to the next stage in your research, when you identified a few candidates, you may want to do a screening in a cell-based assay. So we offer the OX40 and F-kappa beta luciferase reporter HEC293 cell line. So basically, we have introduced the OX40 receptor units into the cell line. Once the ligand is bound, you have the triggering of the signaling pathways that end up with an F-kappa B response element triggering luciferase expression. Luciferase, as you know, is a sensitive readout that can be used very easily, both for viability of the cells, but also for measures of interaction and activation of pathways. This has been validated by flow cytometry and in response to the OX40 ligand. So you can see a very nice dose response. So once you incubate your cells with the ligand, you do see activation and expression of luciferase. In addition, it was validated with the antagonist antibody. So you can see here that in the presence of the antibody, you do see a decrease in luciferase expression and response. No need to mention that we have developed a suite of tools that go with the cell line to optimize it and maximize the results for you, such as claw media, growth media, and assay medium. And we do offer recombinant protein and the antagonist antibody you can use and control in your assays, in addition to our one-step luciferase assay. If you're not interested in measuring luciferase, we have a little bit of a different cell model. In this case, OX40 was introduced, so the readouts will have to be different. There is still an F-kappa beta response element. However, there is no luciferase reporter, so this would be a useful model if you're interested in measuring different outcomes and different types of readouts, or luciferase may interfere with your assays. I want to finalize with introducing to you our custom offers. So if I haven't shown it to you, but you're still interested in the tool, please reach out to us. We're more than open to develop custom projects with you. We cover your discovery from the target protein to the in vivo studies with our solutions from recombinant proteins, antibodies, biochemical assays, inhibitors, cell-based assays, cell models, cell media, supportive cells, virus-based tools. We try to be on top of what tools are needed in the field, but you more than anyone know what you really need. So please reach out. Please talk to our team. All the research is done in-house in the United States, in San Diego, California. So with the good weather to support us on our daily research. We are providing you with customized personal support directly coming from our scientists. As I mentioned, we have a highly scientific team of investigators working with us. We are ISO 9001:2015 certified, and we do provide and perform lot-specific quality control testing. We can target and work with you in protein expression with the choice of tag, label, expression system, and the QC that you required. We do provide cell line development. At the moment, we have more than 30 different cell types. But if there is a cell type that you need and is not included in our portfolio, we surely be happy to work with you on that. And our goal is really to break the bottlenecks in your research, make it quick, and maybe make it fast, make it come to the clinical studies as quickly as possible. So we try to align our service with preclinical drug development, discovery biology, and chemistry. We cover small to large. So if you need 100 micrograms, we're here for you. But if you need 100 megs, we're also here for you. We provide support in process development, scaled up validation, and a number of other types of tools. In conclusion, I think I hope to have shown you that inflammation is indeed a very crucial field. It's a very important in our daily lives, and any imbalance in this response can lead to debilitating diseases, and some of this can be deadly. And if not deadly, they have a severe impact in the quality of life of these individuals. The pharmaceutical companies have been investing in this field heavily in the last years, and I think that's no surprise in view of the field and the number of diseases we're talking about. Three of the major targets that have been investigating are TSLP, TL1A, and OX40. The pathways are complex. They still need a lot of work to be elucidated, understood, and manipulated as best as possible for us to develop new drugs and effective drugs. We have the field has moved very far, so there's multiple strategies that have been used, but there's also a lot of it still to be discovered. And BPS hopes to be your partner in that and cover your discovery workflow from beginning to end, and provide you with the optimized tools at each step, including your cell culture workflow. Thank you very much for your attention. And if you have any questions, please feel free to go ahead. I'll stop sharing.
Thank you very much, Vanda. That was really awesome. It's really comprehensive, actually, the amount of product ranges that we cover, given but specifically for those topics. Is there anything else kind of outside of that you guys can provide to the customer? I mean, you've also talked about, the custom product ranges and stuff like that. Is there anything else that BPS are working on, particularly in those areas that you're planning on offering?
So at the moment, we still want to expand this portfolio. We also want to expand a little more broad to other molecules involved in inflammation. This has three of the main ones, but we are trying to expand our portfolio. I haven't shown, but we also have lentivirus that we can help and support the customers with if they need to express. If they have a cell line and they want to express one of these molecules, we can provide lentivirus, AAV. One of the areas of also quite interesting for us that we hope to expand into is the inflammasome. That's a big word for me to say, but it's a very interesting field, and we're now trying to develop more and more tools to cover that. And I think, as you know, we're open to any type of custom project. We do our best to keep up with the research, keep up with what's being in clinical trials and published, but really the customers know what they're doing behind the scenes, and so we're here to support them as needed. And I don't know if Susan or Lillian want to, bring up any specific product they have in mind or would like to mention, but feel free, Susan and Lillian.
I mean, we do have advocates for what is it now? The can't even remember it. NLMP, NLRP. I get the name of it now. NALP3. Okay, it's one of the inflammasome proteins. So I know we do have some advocates and that we've developed products in that area, but it's something that we're still trying to expand into a little bit.
Yeah. We have a
Right.
lot of products on the CAR T and the immune side. You know, we have PBMCs, we have cytotoxicity assays, we have a lot of cell lines that are designed as cytotoxicity targets. So we're really trying to cover a lot of the fields of the immunology, in particularly inflammation. I think we now are going to put some effort into the inflammasome and continue developing tools on new molecules for inflammation.
Good to know. Is there any other questions before we end? Maybe we could do another webinar later in the year, Vanda, particularly for CAR T cell product ranges, because there is an awful lot of researchers in Australia who work on CAR T cell products.
Yeah, we have a very nice workflow setup. You know, we try to cover from beginning to end on the CAR T side again, you know, starting with a very basic research towards more of the clinical side. So we would go from the gene sequencing to the CDX module. So I'll be very happy to share the type of products we have on that area with you and your
That'd be
team.
great. Fabulous. Okay, on that note, it's just past 11. I'll end this meeting. Thank you very much for attending. I really appreciate it. I'll be in touch with everybody who's attended today just to follow up and see if you need any further information. And thank you very much, Vanda, for a really comprehensive presentation. I really appreciate it.
Thank you. Thank you very much for having me, and have a good day.
Thank you.